Hi, everybody. Today we are going to talk about receptors. So if you remember, when we talked about cell signaling, we said there were three parts. There was reception, and then there was transduction, and then there was a reaction, something that happened. And today's lecture is about reception. Now, if you remember yesterday, there were two types of receptors. Hopefully you remember there were membrane receptors and cytoplasmic receptors. We're going to talk about the cytoplasmic receptors first. Things that are small or hydrophobic. So ligands that are small or hydrophobic, they can diffuse across the bilayer through that plasma membrane, and they can enter the cell on their own. So things that are small or nonpolar are going to use cytoplasmic receptors. Estrogen, for example, this is a steroid hormone, and that means it's lipid soluble. So estrogen can easily diffuse across the plasma membrane and enter the cell, and therefore it is going to react with things using a receptor protein that is present in the cytoplasm. Here, I'll move this out of your way so you can see. Here we have an example of a cytoplasmic receptor. So here we have a steroid hormone. It goes right through the plasma membrane. And notice what's going on here. We have the receptor, but the receptor is not active. And when the steroid hormone binds to the receptor in the cytoplasm, it's going to change the receptor's shape, and it can now enter the nucleus. So before it couldn't enter the nucleus because it had the wrong shape. Now it can. It's going to enter the nucleus, and typically what these receptors do is they are going to transcribe the DNA. Right? That means they're going to read the DNA and they're going to copy the message down in order to get it ready to make a protein. This is going to be a way of making a protein or an enzyme or some cell component. The steroid hormone activates a receptor. The receptor can go in the nucleus. So the receptor for the hormone cortisol, for example, is normally bound to a chaperone protein. We'll take a look at that in a minute, which blocks it from entering the nucleus. Binding of the hormone causes the receptor to change its shape so that the chaperone is released. And then this allows for the appropriate conformational change so that it can enter the nucleus. Here we have an example of cortisol doing just that. Cortisol is small and nonpolar, so it can go right through that membrane. Here, it attaches to the receptor, so that is the receptor. Now, before it attaches, the receptor is attached to this chaperone protein, and it's the wrong shape, so it cannot enter the nucleus. But once the cortisol attaches, once the ligand attaches to the receptor, it causes a conformational change such that the chaperone protein is kicked off, and now the receptor can enter the nucleus and begin transcribing DNA into RNA. And that RNA can, at some point, be translated into a protein. So here we have yet another example of the steroid hormone going through, kicking off the chaperone protein, causing transcription, and then we end up with a new protein being made as a result of the presence of that steroid hormone. All right, and that is where we are going to stop with cytoplasmic receptors. And now we need to move on to membrane receptors. There are three main types of membrane receptors that we are going to go over. So if you are a ligand that is large, polar, then you can't do what those hormones did. You cannot pass through the membrane. So you need a membrane receptor. Insulin, for example, is a protein hormone that can't diffuse. So insulin is something that is going to have to use a membrane receptor. We have three different types: ion-gated channels, protein kinase receptors, and then G proteins. And we're going to talk about each one. So first, a question: what type of ligands would use a cytoplasmic receptor? And then, what kinds of ligands would use a membrane receptor? Type in your answers now. All right, so we are going to move on and talk about our first type. We are going to talk about the ion-gated channel receptors. So the ion-gated channels are going to be channels that let ions through. So sodium ions, potassium ions, calcium or chloride ions. So here we have an example of it working. So we have a ligand, and we have over here, we have our channel. The channel is closed. So notice that we have ions, and these ions could use the channel, but they can't right now because the channel is closed. So our ligand, this larger polar ligand, is going to attach to the reception site, and that's going to cause a conformational change, opening up the channel and allowing ions through. So ions are then going to act as a signal, and they are going to do something later. So they can cause some cellular response down the line. Right? Each type of ion channel has its own signal. So one signal does not work for every ion channel. We need specific signals for specific ion channels. The stimuli that could act as a signal, it could be a ligand, but it could also be light. It could be sound. It could be an electrical charge, which is what happens in our nervous cells. Or it could be a chemical ligand such as a hormone or a neurotransmitter. So there's many ways that these particular channels can open. So here we have a ligand. It attaches to the binding site. It's going to open that channel, and now these ions can diffuse. Remember, this is passive. They can diffuse through and get into the cell. This is actually one of the ways that our nerves work. Here we have an example of acetylcholine. So acetylcholine is a neurotransmitter, which means it sends messages between neurons. When two of them, in this case, attach, so we have to have two, it is going to open up the channel. Here we have the sodium ions that are present, and they are able to go through. Sometimes these channels can be aligned with negatively charged amino acids, attracting positively charged ions through. And this is going to lead to a muscle contraction because acetylcholine is a neurotransmitter for muscle cells. All right. And we have a few examples. We'll go over those later. All right. The acetylcholine receptor, which is located in the plasma membrane, is an example of a gated ion channel. This receptor protein is a sodium channel. So when acetylcholine binds to the protein, it lets sodium ions through that channel. And that is going to continue a nerve impulse to allow the muscles to move. When two molecules of acetylcholine open the receptor, it is enough time, even though it's a very short time, it's enough time for tons of sodium to actually get in. And that increase of sodium ions in the cell is going to initiate a series of events that results in a muscle contraction. So we have the reception, the acetylcholine, two of them binding, opening, letting the sodium in, and that sodium is then going to cause a bunch of things to happen in the transduction phase, and the result is a muscle contraction. Here is your lecture check on your own. Please summarize how an ion-gated channel would work. Use an example, or you can just discuss in general. Okay, so moving on to receptor number 2, protein kinase receptors. These are interesting, a little more complicated than an ion channel. And protein kinase receptor is going to be one that involves a kinase. So think about, all right, what's a kinase? You should already know that a kinase is going to phosphorylate something. So these receptors are going to act as enzymes, and they're going to catalyze the transfer of a phosphate group from ATP to a protein. And we're going to refer to that protein as a target protein. So let's find out how this works. Okay, so we'll look at a picture first. First of all, we have the protein kinase receptors. So notice they're separated a little bit, but not by much. We have a signal molecule. This is the appropriate signal for the receptor. There's actually an alpha helix shape in the membrane. That's not too important, but just some connection there. When we have two signals and they both attach to the receptors, it is going to cause a conformational change. And that conformational change is going to occur so that it kind of forces these two subunits together to make a dimer. Two parts have come together for one. This is now an active protein. There are all of these tyrosines here that are going to act as a kinase. So these are going to each react in step 3 with a ATP, and the ATP is going to phosphorylate the tyrosine. And now the tyrosine can act as a kinase. And when a specific protein matches up with it, it is going to phosphorylate that protein. Phosphorylating the protein is going to cause a conformational change of that protein, which is going to then activate the protein, and that protein can go on to do something. So this protein was inactive, not really working the way it should, meets up with the kinase phosphorylates it, now it has its phosphate attached, now it can do whatever it needs to do. All right, so here is another view of that happening. We have the receptor. The receptor is receiving insulin. It is now going to form a dimer. It is active. It has been phosphorylated. So here is the inactive protein. The inactive protein meets up with that kinase, picks up a phosphate, and boom, now it is active. And now it can do whatever it needs to do, whatever that response is. We'll worry about that at a later time. Right? This is a protein kinase receptor. And we're going to be looking at some of these examples later. We're going to skip this for now. So our example for the tyrosine kinase receptor is the insulin example. Insulin is going to be made by the pancreas. So insulin is basically going to go around and it's going to try to lower our blood sugar by bringing that sugar into our cells. So if our blood sugar is high, let's say you ate a bunch of candy, insulin is released, and insulin is going to find the appropriate cells in your liver or in your muscles, and it's going to attach to the receptor. The receptor is then going to activate a signal, and that signal is going to bring sugar into our cells for storage, keeping them out of the blood. All right. The insulin receptor autophosphorylates, so it phosphorylates itself. We saw that in the picture. And then it's going to catalyze the phosphorylation of a protein. These proteins may initiate many responses, including what insulin does, the insertion of glucose transporters into the plasma membrane. Remember, this brings glucose into the cell and out of the blood. So it lowers our blood glucose levels. All right, lecture check. Take some time to describe how a kinase receptor is going to work. You can speak about it in general, or you can use the example of insulin. Okay, our third type of receptor is a G protein receptor, and this is maybe the most complicated one, but it's also very common, and we're going to look at a number of examples of this. So there are three main parts of a G protein coupled receptor. There is the G protein coupled receptor, which is bound to the membrane. It's usually like a transmembrane protein. We have the G protein, which is made up of a few subunits, and it is attached to the other part. And then we have an effector protein. Okay, we're going to watch some videos on this at a later time. So first of all, let's look at the G protein coupled receptor. So which is this? It's the part, the transmembrane part. It contains 7 transmembrane regions that pass through the bilayer. We have a ligand binding site on the outside. And when a ligand is bound, it is going to change the shape of the cytoplasmic region. Just like every other membrane receptor, when the ligand binds, the cytoplasmic side is going to change. It's going to undergo a conformational change. And this is going to affect the G protein, which is kind of waiting right around where I have the pointer. So we're going to go over the verbal description first, and then we're going to see the picture to make sense of all of this. So a G protein has 3 subunits, and they can bind something called the GDP and GTP. You may think those are similar to ADP and ATP, and you would be correct because they are similar to ADP and ATP. These things are going to act in a very, very similar way where they can lose a high energy phosphate and then reattach it. It's just it's not adenine, it's guanine instead. It works in the same way as ADP and ATP. A G protein has 2 important binding sites, one for the receptor and then one for GDP and GTP, and we'll look at a picture of that in a minute. The inactive G protein binds GDP to its subunits if it's inactive, and it exchanges that for GTP when it is activated. So the activated G protein has GTP, highly energetic. The G protein that is inactive has GDP. At the same time, the ligand gets released. So when that G protein is activated, the ligand goes away so that it can be used again. All right, and at that point, the G protein is going to move and it's going to encounter the third type of protein, the effector protein. The effector protein is just what the name implies. It is going to cause an effect in the cell. So when the G protein hits the effector protein, something is going to happen, and we'll worry about what happens later. After binding to the effector protein, the GTP is going to get rid of that phosphate and it's going to become GDP, and now the G protein is inactive. And everything basically goes back to the way it was at the beginning so that this can all happen again. So what does this look like? So here we have a signal. So we have some sort of polar or large hormone, and it attaches to the G protein linked receptor right here. When this happens, so here's our inactive G protein. Notice it's inactive, so what does it have? It has GDP. All the way on the right is our effector protein. When the ligand attaches to the receptor, the cytoplasmic side of the receptor is going to activate. It's going to change shape, conformational change, and it is going to interact with our G protein. This activates our G protein, and our G protein kicks away the GDP and attaches a GTP. So notice what's happening here. It is not simply adding on a phosphate. That is not happening. It is getting rid of GDP in its entirety, and it is taking a totally new GTP. When that happens, it is going to cause the movement of the G proteins. The G proteins, like a shuttle, it is going to move. And notice in the picture how it has moved. Part that has the GTP keeps going and hits the effector, activating the effector protein. The effector protein had just been sitting there, but now it is active. The effector protein can work very quickly, and it can start to cause a change in the cell. And notice down here it says amplification. That's because it is going to cause a big, big change very, very quickly. And we'll find out some examples of those changes. So this is a G protein. Now, after this has happened, after we have activated the effector protein, we lose a phosphate. And if we lose a phosphate, it's going to become GDP. And if we become GDP, everything is going to go, like the G protein is going to inactivate, and it's going to all go back to normal. Notice what else happened. When the G protein left, it changed the shape so that the ligand got kicked out. This is kind of resetting, allowing it to be reused again. So now it is time to explain how a G protein linked receptor is going to work. Take some time and work on that now. And that is all for today. We're going to start modeling each of these types and reviewing them. All right, and we will watch videos later. So otherwise, let me know if you have any questions.